and welcome back to Learning with Isha. We are in the plant growth and development series and we are looking at what we know as phytohormones. We've already looked at 3 plant growth promoters, auxins, gibberellins and cytokinins. We've looked at another plant growth hormone that falls in between the category of promoter and inhibitor. It is known as ethylene. Today we move on to the last category, that is the inhibitor of plant growth, and the name of this substance is abscisic acid. Well, abscisic acid is not only known to inhibit the plant growth but also inhibit the metabolism in plants. This abscisic acid was discovered by 2 separate groups of scientists. Let's look at the discovery. The first group was 2 scientists, Carnes and Adicott. While they were observing how the cotton balls were shed off from the cotton plant, after the cotton ball was ripe and the cotton was ready, how it was falling off from the plant. They found out that there was a substance known as Abscisin II that was responsible for this shedding. Another group of scientists called Waring and Carnforth observed that the buds of the plant did not open up to form flowers. They were dormant due to the presence of a chemical. And this chemical that was inducing dormancy was very rightly named as Dormin. Later studies illustrated that both these chemicals, Abscisin II and Dormin, were in fact the same chemical, and together they came to be known as abscisic acid. Now let's move on and look at the functions of abscisic acid. The first most important function of abscisic acid has to be in inducing dormancy. We already know that it was known as dormin because of its ability to induce dormancy. So where is it going to induce dormancy? Well, both in seeds and in the buds of the plant. Now let's talk a little bit about seed germination. A seed has some reserve food material in it. It is going to store some starch or some protein. Now this protein or starch is going to be broken down by enzymes. Let's say if I talk about starch, it would be broken down by amylase enzyme. And if I talk about proteins, proteins are going to be broken down by proteases. Well, these enzymes are nothing but proteins. And we know these proteins are formed from the mRNA by the process known as translation. So, if the translation process is hindered, there are not going to be any proteins. That is exactly what abscisic acid does. In a seed, it is not going to allow the translation process. In the absence of translation, these enzymes are not going to be formed. If there is no amylase, no starch would be broken down. And if there is no starch breakdown, the seed is not going to have enough energy to be able to germinate. So that is how exactly abscisic acid induces seed dormancy. Similarly, it will also induce the bud dormancy. It will not allow the bud to open up and form the flower. Well, how is this process useful for the plants? Now let's say if the seed germinates in highly unfavorable conditions. The seed was supposed to germinate in a pleasant weather and it germinated in a very harsh winter climate. What would happen? The seedling would immediately die due to the harsh cold. That is what the abscisic acid is going to give protection against. Now the next function is that it induces stress tolerance. It is going to allow the plant to withstand stressful conditions. Let's say the heat is very severe. There's going to be a lot of water loss through transpiration. And we know transpiration happens through a structure that we know as stomata. So now to prevent the water loss, we need to close the stomata. How is this closure of stomata going to happen? This is going to be induced by abscisic acid. Let's go back a little to the structure of stomata. The stomata is found on the lower epidermis, as in the lower surface of the leaf. The stomata is basically a pore through which the transpiration happens. That is, the water evaporation in plants is going to happen. This happens due to the presence of two kidney bean shaped cells around the spore that are known as guard cells. Well, how does this stomata close? What happens is these guard cells accumulate potassium ions inside of them and also accumulate malic acid. Due to the accumulation of solutes, the concentration of solutes is going to be high inside the guard cells. So there is a high concentration of solutes here. We know that water moves in from lower concentration to a higher concentration of solutes. So definitely water is going to move inside. Due to the movement of water inside the stomata, the guard cells become turgid and they open up. That is how the stomata opens up. Now our aim here is to cause the closure of stomata. So the abscisic acid is going to exactly reverse all of it. Abscisic acid is going to cause the leakage of malic acid. And secondly, it is not going to allow the uptake of potassium ions. As a result, the concentration of solutes inside the guard cells is going to become low. Due to this, the water will now move outwards and the guard cells are going to close. That is how abscisic acid causes the closure of stomata. Abscisic acid also has a role in the senescence process. Senescence is actually the aging process in plants. So how would this senescence happen and where does it happen? Normally the senescence is of the leaves. Now if the leaf has to age, what is going to happen? Number one, the chlorophyll is going to be lost. The chlorophyll is going to be lost. Now, if there is no chlorophyll, there is a breakdown of chlorophyll, there is going to be less photosynthesis. Now, if there is less photosynthesis, less food is going to be available to the leaf. Besides that, this abscisic acid also changes the metabolism of proteins and nucleic acids in the leaf. Due to the lack of proteins and nucleic acids, there is enhanced aging of the leaf. That is how abscisic acid causes senescence. Now, eventually, if there is aging, this aged part of the plant is going to fall off. This fall off of the aged part of the plant, let's say it could be the leaf or it could be the fruit, is known as a process known as abscission. That is the shedding. How does this abscission process take place? Now, this abscission is not a one-day event. In fact, it is a series of events. When a young leaf is produced, so this young leaf has a very high concentration of plant growth promoters, auxins, cytokinins and gibberellins, but mainly auxins. With time, the senescence process happens, the leaf ages and the concentration of auxins goes down in the leaf. Now, as the concentration of auxin will go down, there would be accumulation of abscisic acid. Now, this abscisic acid accumulation in the leaf causes several things. Number one, in the petiole of the leaf, a layer is formed which is known as the abscission zone. This layer is known as the abscission zone. What happens in the abscission zone is that there is disintegration of the cell wall. Due to the disintegration of the cell wall, there is going to be lack of strength. Due to this lack of strength, the leaf or the fruit is going to be shed off from the plant. That is how the abscission process takes place. Now let's talk about some more facts about abscisic acid. It is also known to be antagonistic. It is known to be antagonistic to gibberellins or gibberellic acid. What is an antagonist? Antagonist is something that works against something, reverses the effects of something. Let's say gibberellic acid is known to cause seed germination, and we know that abscisic acid is known to induce seed dormancy. There are several other functions because of which abscisic acid is known to be antagonistic to gibberellic acid. Also, it is known as a stress hormone. We've already discussed it as it increases the plant's tolerance to both biotic and abiotic stress conditions. Let's say if there is drought, it is going to save water by the closure of stomata. Well, this concludes our plant growth and development series for now. We've not only talked about plant growth, but about the various hormones that either promote the growth or retard it. Now, please let me know in the comments section if you have suggestions for more videos. Don't forget to subscribe to my channel. See you on Saturday with another interesting episode. Bye.